Welcome again. Today we will be talking about cell respiration. Every cell produces its own ATP by a process called cell respiration. Carbon compounds such as glucose and fat are carefully broken down, and the energy released by doing this is used to make ATP. Cell respiration is defined as the controlled release of energy from organic compounds to produce ATP. Cell respiration can be broken down into 4 steps. These steps are glycolysis, link reaction, Krebs cycle, and chemiosmosis. The first step of cellular respiration is glycolysis. This occurs in the cytoplasm and can be broken down to further steps, which are phosphorylation, lysis, and oxidation, which leads to ATP formation. The first molecule that is needed in glycolysis is a 6-carbon compound called glucose. 2 phosphate groups are added to a molecule of glucose, and this forms hexose bisphosphate. Adding a phosphate group is called phosphorylation. The 2 molecules of ATP provides the phosphate groups. The energy level of hexose is raised by phosphorylation, so this makes hexose bisphosphate extremely unstable, and this leads to lysis, which is splitting of hexose bisphosphate into 2 molecules of triose phosphate. 2 atoms of hydrogen are removed with the help of 2 NAD, which this process is oxidation. The energy released by oxidation of each triose phosphate molecule is used to convert 2 ADP molecules to ATP. The end product of glycolysis is pyruvate. So what you get in the end after glycolysis is 2 pyruvate molecules and 2 ATP molecules. So it's not a very efficient process in terms of formation of ATP. Next step after glycolysis is link reaction. The 3 steps in link reaction are oxidation, decarboxylation, and the addition of coenzyme A. While link reaction occurs in the mitochondria, as the pyruvate formed from glycolysis is absorbed by mitochondrion. Enzymes in the matrix of the mitochondrion remove hydrogen and carbon dioxide from pyruvate. The removal of hydrogen is called oxidation, and this is when hydrogen is accepted by NAD. The removal of carbon dioxide is called decarboxylation. The whole conversion is therefore called oxidative decarboxylation. The product of oxidative decarboxylation of pyruvate is an acetyl group, which is attached to coenzyme A to form acetyl coenzyme A. After the link reaction follows the Krebs cycle. The Krebs cycle occurs in the matrix of the mitochondria, and the steps that are involved in the Krebs cycle include oxaloacetate is converted into citrate, and there is also decarboxylation and oxidation reactions. The cycle helps with the regeneration of oxaloacetate. And there's also substrate level phosphorylation, which helps with the formation. Acetyl groups from the link reaction are fed into the Krebs cycle. In the first reaction of the cycle, an acetyl group is transferred from acetyl coenzyme to a 4-carbon compound called oxaloacetate to form a 6-carbon compound called citrate. Citrate undergoes oxidation with the hydrogen carrier NAD and decarboxylation. This makes a 5-carbon compound. Oxidation and decarboxylation happens again to form a 4-carbon compound. With the 4-carbon compound, substrate level phosphorylation happens, which forms ATP. Oxidation has once again happened with the hydrogen carrier FAD to form FADH. Oxidation is once again happened with the hydrogen carrier NAD to form NADH, and NADH is carried out. This helps citrate be converted back into oxaloacetate and is fed back into the Krebs cycle. So in summary, there are 2 reactions of decarboxylations, meaning that the carbon dioxide is removed in 2 of the reactions. The carbon dioxide is a waste product and is excreted together with the carbon dioxide from link reaction. Hydrogen is removed in 4 of the reactions, and this reaction is oxidation, as I mentioned before. The final stage of cellular respiration occurs in the electron transport chain, or the cristae. The NADH and FADH that came from the oxidation reactions in the Krebs cycle supplies two electrons to the first carrier in the chain. The electrons come from the earlier oxidation reactions. As the electrons are passed along the chain from one carrier to the next, they give up energy. Some of the electron carriers act as proton pumps and use this energy to pump protons against the concentration gradient from the matrix to the mitochondrion to the inner membrane space of the mitochondrion. And FADH also feeds electrons, but then this is slightly in the later stage than the NADH. At the end of the electron transport chain, the electrons are given to oxygen. Um, this happens in the matrix on the surface of the inner membrane. At the same time, oxygen accepts free protons to form water. The use of protons in this reaction contributes to the proton gradient across the inner mitochondrial membrane. Energy released from as electrons pass along the electron transport chain is used to pump protons across the inner mitochondrial membrane into the space between the inner and outer membrane, and this includes the space inside the cristae. A concentration gradient is formed, which is a store of potential energy. Now, this concentration gradient allows for the diffusion of protons back into the matrix from the inner mitochondrial space to the matrix. This is where ATP synthase comes in. ATP synthase, also located in the inner mitochondrial membrane, allows the protons to diffuse back, and the ATP synthase uses the energy that the protons release as they diffuse down the concentration gradient to the proton to produce ATP. The generation of ATP using energy released by the movement of hydrogen ions across the membrane is called chemiosmosis. This is the mitochondria where the cellular respiration happens. There's the matrix, ribosomes, intermembrane space, cristae, and external membrane, all designed so that maximum ATP can be formed.